Hello, my name is Camille Kaur, and I am a medical student from the University of Rochester. I am honored to be here today to do a brief but deep dive into Batten disease that I hope will empower you with a basic understanding of this condition that has found its way into you and your family's lives. I don't have any conflicts of interest to disclose. So first, today we will cover a brief introduction to Batten disease. We'll go over exactly what a lysosome is and what it means that Batten disease is a lysosomal storage disorder. We'll start to unpack the basics of the genetics of Batten disease, including how it runs in families. We'll talk about genes, chromosomes, DNA, RNA, and proteins, and how changes in your genes can cause some people to develop Batten disease and others to be unaffected. We'll discuss some of the ways different forms of Batten disease are similar and some of the unique features that set them apart from one another. Once we've covered a broad overview of Batten disease as a group of disorders, we'll discuss several different types of treatment that are possible for the different forms of Batten disease. We'll talk about symptomatic care, enzyme replacement therapy, and gene therapy. Finally, we'll wrap up by discussing some of the therapies that are currently being developed and researched. So today we're going to discuss a group of diseases that go by a lot of different names. The most common name is Batten disease, but the scientific term is neuronal ceroid lipofuscinoses, or NCLs. The different forms of Batten disease sometimes have more specific names, but they're commonly known by their number, CLN1, CLN2, CLN3, etc. So whenever you hear about Batten disease or the NCLs, we're talking about the group of disorders. And when we say CLN2 or CLN3, we're talking about one form of Batten disease. The NCLs are part of a group of diseases called lysosomal storage disorders. Lysosomes are a part of the cell in our bodies that normally gets rid of waste. And when the lysosomes don't work properly, waste products begin to accumulate or build up in the cell. What these diseases have in common is that a certain waste product, called ceroid lipofuscin, builds up in the cells, which you can see in the images on the screen that we'll discuss in a little bit. Next, Batten disease is a rare disease, and only about 2 to 4 babies out of every 100,000 born in the US are diagnosed with it. There are 13 different forms of Batten disease. There are different key features that they share in common, but also a number of features that set them apart from one another. One of the main differences between the different forms is the age when they first start. Some of them start as early as infancy, some start in early or in late childhood, and some not until adulthood. Some of the other ways that you can distinguish between the different forms are by looking at the specific gene that is involved, the order that different symptoms occur, and the rate of progression. So first, I'd like to talk a little bit about lysosomes and why they are relevant to Batten disease. So just like the human body has different organ systems to do different jobs, the heart and the cardiovascular system pump blood throughout our body, the digestive system absorbs the nutrients that we need to fuel our body. Each of our cells has a number of different organelles, or little organs, which each serves a different purpose to help keep our cells running properly. For example, the nucleus is the control center or brain of the cell. On the left side of the screen, it's the purple ball in the middle. This holds the cell's genetic information, which is the set of instructions that say how our cells are supposed to work. Certain diseases happen when one of those organelles in the cell doesn't function properly. And in Batten disease, the organelle that doesn't function properly is called a lysosome. So lysosomes' role in the cell is to remove waste. They're sometimes known as the garbage collectors or the recycling centers of the cell. The lysosomes contain a really acidic environment, sort of like the really acidic environment that's in the stomach, as well as different enzymes that help to break down that waste. There are a lot of different steps involved in the way that the lysosome breaks down waste, and just a few of these steps are shown in the image on the right. But if any one of these many different steps doesn't work properly, the lysosome isn't able to accomplish its goal of breaking down all of the cell's waste products. And as a result, waste products start to build up in the cell. In Batten disease, when the lysosomes don't function properly because of a problem with one of those many steps, there's buildup of a specific waste product called ceroid lipofuscin. Ceroid lipofuscin accumulates especially in the nerve cells, or the neurons. In other lysosomal storage disorders, waste products build up in different organs, like the liver, which causes those diseases to look a little bit different. Batten disease causes neurological symptoms because the ceroid lipofuscin is building up in those nerve cells. Something that's pretty interesting is that ceroid lipofuscin can also build up in cells with normal aging, even in people without Batten disease. This is probably because our lysosomes stop working quite as well when we get older. Ceroid lipofuscin itself doesn't directly damage the neurons, but it's a marker that things are not working properly. Some people call this a biomarker. That is why when we see ceroid lipofuscin in nerve cells in a young person, we begin to suspect Batten disease, since we know that their lysosomes are still supposed to be working pretty well. If you break down the word ceroid lipofuscin, you get ceroid, which means wax, lipo, which means fat, and fusin, which means color. So ceroid lipofuscin is basically a collection of waxy fats and proteins that are colored under the right kind of light. And one of the ways that we test for different forms of Batten disease is looking for specific patterns of buildup of ceroid lipofuscin in cells. We can collect a tissue sample or a biopsy from a patient, apply different dyes and stains to it, and then look at the tissue under a microscope to see whether we can see any of that ceroid lipofuscin. The way these different stains work is by attaching to specific proteins and fats that we know are part of ceroid lipofuscin. In the top 4 images, a researcher from Helsinki, Dr. Haltia, shows the ceroid lipofuscin that you can see by using different stains. We can also look at tissue samples under an especially powerful microscope called an electron microscope. This is helpful to see different patterns that can help determine which form of Batten disease someone has. In the bottom 4 images, Dr. Haltia shows that what some of these patterns look like. One of the specific patterns that you may have heard of is fingerprint deposits, which are seen in CLN3 disease. If you look closely at the 3rd image in the bottom row, you may be able to see exactly what those look like. I think they look a lot like fingerprints. The NCLs are inherited disorders, which means that they run in families. You've probably heard the terms DNA, genes, and chromosomes come up in conversations about Batten disease. I think the image on the right is pretty helpful in understanding how those terms are related. So DNA is the genetic information that serves as a set of instructions to tell our body how to develop and function. DNA is broken up into segments called genes, which are shorter sets of instructions for one specific function or part of the body. Now, in our cells, DNA is coiled up super tightly so that it takes up less space. If you stretched out all of the DNA from all of the cells in a person's body, it would be able to wrap around the entire solar system twice. So you can see why it's so important that it's coiled really tightly. The coiled up version of DNA is what's called a chromosome. Each of us has 46 chromosomes, with one set of 23 chromosomes coming from a mother and one set of 23 chromosomes coming from a father. This means that each parent passes on half of their genetic information to their child. The picture on the left is called a karyotype. This is a picture of a person's entire set of chromosomes. You can see that there are 2 copies of each chromosome, numbered 1 through 22. And then X and Y are sex chromosomes. And whether we inherit 2 X chromosomes or an X chromosome and a Y chromosome determines our sex at birth. The person this karyotype belongs to is biologically male because they have an X and a Y chromosome. Now that we know that DNA is the set of instructions that tells our body how to develop and function, we can start to think about exactly how it does that. Just like the English language is made up of letters that group together to form words, DNA is a genetic code that works like an alphabet to spell instructions to our cells. While the English language, or the English alphabet, has 26 letters, the genetic alphabet only has 4 letters, which we call nucleotides, A, C, G, and T. These nucleotides, or letters, are grouped in 3s to form words, which we call codons. Now, once we have a strand of DNA, which we know now is just a strand of letters, Something called RNA comes in to transcribe that strand of letters into a form that can eventually be made into proteins. So by this point, we have RNA codons, or words. Each of these codons corresponds to one amino acid, which is a building block for proteins. At the end of this whole process, we end up with a strand of amino acids, which come together to make a complete protein, which is our final product. Unfortunately, our body isn't perfect, and sometimes spelling mistakes happen. We call these errors mutations or variants, or sometimes polymorphisms. We have a few ways to proofread and fix errors in our body, but we can't catch them all. Some of these spelling errors end up causing disease. Just like in the English language, some spelling errors can change the meaning of an entire sentence, but some spelling errors are pretty minor, and the sentence can still be read normally. I'm going to talk about 4 different types of spelling errors that can happen to our genetic code and how they affect the DNA. The first type is called frameshift errors. Remember that all of the words or codons that DNA is broken up into are 3 letters long. If a mistake happens where you add or remove 1 or 2 letters, every word after that change gets messed up, since each word has to have 3 letters. These errors tend to be pretty severe. The next type is insertion errors, where a bigger chunk of letters gets added, but in a multiple of 3. When letters are added in groups of 3, like in the top example under insertion, the meaning of the entire sentence changes slightly, but everything after it stays the same. When instead of a chunk of letters, a stop signal gets added, kind of like a period in the middle of a sentence, the rest of the sentence disappears. This is the bottom example under insertion. When these errors, they also tend to be pretty severe. The next type of error is a deletion error, where a bigger chunk of letters gets deleted, but in a multiple of 3. When letters are removed in groups of 3, the meaning of the sentence will change slightly, but again, everything after it will stay the same. And the last main type of spelling errors that I'll talk about are substitution errors, which is when a single letter gets changed to another letter. When this happens, the meaning of the sentence may or may not change. So in the example on the screen, if the C in cat gets changed to a K, we'll still read the sentence as the cat and the dog ran. But if we change that C to a B, the meaning of the sentence changes, and we now have a bat and a dog ran. Depending on whether the meaning of the sentence changes, so to speak, a spelling error or mutation may or may not interfere with those instructions a cell needs to work properly. Sometimes an error changes just enough that the instructions can still be understood, and sometimes it makes the instructions completely uninterpretable. This is why we sometimes need even more information than just genetic testing to figure out whether someone has Batten disease. They might have spelling errors, but not severe enough to cause the disease. These errors that we're not sure about are sometimes called variants of unknown significance. It's also important to note that all of the different types of errors or mutations occur in all forms of Batten disease. Some of these variants are common, sometimes in certain populations. For instance, you may have heard of the Finnish variant or the Turkish variant. In CLN3 disease, there's what's called a common deletion that is present in 70% of patients with CLN3 disease, but the other 30% have different types of variants. What these variants have in common is that when you have a spelling error that changes the meaning of the genetic code, the lysosome is not able to function properly, which is what causes a person to have Batten disease. For the most part, Batten disease has what is called autosomal recessive inheritance. When families learn that Batten disease runs in families, some families might wonder why they didn't know that Batten ran in their family earlier. And this is because of this autosomal recessive inheritance. I'm going to unpack a little bit more about what that means and how it might affect you and your family. We learned earlier that every person has 2 copies of each gene because they have 2 copies of each chromosome. As long as a person inherits at least 1 copy of the Batten gene that works right, they will not develop Batten disease. These would be the white boxes on the screen. People who have one copy of the gene that works right and one copy that doesn't are called carriers in the light blue. And then people who have two copies of the gene that don't work right will have Batten disease. Let's talk a little bit more about carriers in light blue. Carriers don't have Batten disease themselves, but if two carriers of Batten disease have a child together, they can potentially, but not definitely, have a child who gets two copies of the gene that don't work right. This would mean that that child would have Batten disease. This happens about 1 in 4 times, or 25%. But even between those two carrier parents, they still are going to have a 2 in 4 chance, or a 50% chance, of having a carrier child, just like themselves. And finally, they'll have a 1 in 4 chance of having a child who does not have Batten disease and is not a carrier. And this is why, in any given family, one child might have Batten disease while another does not. There are about 13 different forms of Batten disease, and each form really is its own unique disease. Some of the things that put these diseases under the Batten disease umbrella are the buildup of the waste product, ceroid lipofuscin, in nerve cells. All forms of Batten disease have neurodegeneration, some form of eye disease, seizures, and some type of movement disorder. But there are also a lot of things that make each form unique. Different forms might have different symptoms beyond the ones in yellow on this slide. Like we discussed earlier, some forms begin in infancy, some in childhood, and some not until adulthood. For the symptoms that all of the forms have in common, in yellow, in different forms, they might start in a different order. In one form, seizures may come first, while in others, it might be vision loss. Different forms of Batten disease progress at different rates, some more slowly and some more rapidly. And finally, different forms have different cell markers and patterns, like the fingerprint deposits we saw earlier, which are present in CLN3 disease. The first step in diagnosing Batten disease is for someone to suspect it, and then for us to figure out what form of Batten disease someone has. To do that, we have several different tools that we can use. The first is genetic testing, which allows us to figure out exactly which spelling errors a person has in their genes. Currently, most diagnostic testing is genetic, but we also need to know other information to help us interpret those results, thinking back to those spelling errors, which didn't necessarily change the meaning of the sentence. Sometimes we find a spelling error and we want to know if it is significant or not. The next method is biochemical testing, where we answer the question, what part of the lysosome doesn't work? We can do this by testing enzyme function or by collecting a tissue biopsy, sometimes of the skin or of the eye, and then looking at it under a microscope. The next two pieces of information that are important for making a diagnosis are age of onset and clinical symptoms, since we know that these look a little bit different in different forms of the disease. Now, our way of making a diagnosis has changed pretty dramatically in the last few decades. 30 years ago, we really based most of our diagnosis on age of onset. Then 20 years ago, we started to rely a little bit more on biochemical testing. And then in the past 15 years or so, genetic testing has become the gold standard. And sometimes it is sufficient on its own. But even now, we may need additional information to tell us whether a spelling error in the genetic code is causing Batten disease or not. Next, I'm going to transition to talk a little bit about the different strategies that we have in the present and in the future for treating Batten disease. I'll talk about 3 different approaches, though there are some others beyond these. We'll cover symptomatic care, enzyme replacement therapy, and the emerging field of gene therapy. The first method that we have is to treat the symptoms of Batten disease. It's important to remember that these can be valuable tools to work on one aspect of Batten disease, but they don't treat the underlying disease itself. These are things like anti-seizure medications, medications to treat movement problems or to manage mood and behavior, different supportive therapies like physical therapy, occupational therapy, low vision therapy, and helping someone with feeding. Finally, we have palliative care, whose focus is to maximize someone's quality of life. The next treatment method is called enzyme replacement therapy. Some forms of Batten disease are caused by an enzyme that is missing because it didn't get made properly. Because these enzymes are soluble in water, they can travel through the brain when they're given as treatment. The video that's playing on the slide is showing how water flows through the brain, with different colors representing different directions. This shows us that anything that we can dissolve in water should be able to travel throughout the brain to different areas. For enzyme replacement therapy, if we give a patient the enzyme into the spinal fluid, it can travel to different parts of the brain. One example of this is Brineura, which is approved for CLN2 disease. Unfortunately, this treatment only works for CLN2 disease because it is specific to the enzyme that is missing in that form. The next treatment method is gene therapy. We learned earlier that patients who have at least one working copy of the Batten gene typically do not have the symptoms of Batten disease. This means that it may be possible to give patients a new working copy of the Batten gene. We hope that by doing this, we can prevent someone from showing symptoms of Batten disease, just like those carriers who have one working copy of the Batten gene and one non-working copy, but don't have Batten disease. In the picture on the right, you can see a working copy of the Batten gene getting added to the cells of a person with Batten disease. While this is still an area of ongoing research and there are no currently approved gene therapies for Batten disease, it's a really exciting area that's emerging with ongoing research. There are two major forms of gene therapy called in vivo and ex vivo. So in vivo gene therapy, which is represented on the left, is where we take a copy of the gene and package it into what we call a vector. I think about it like a spaceship. And basically then that vector or that spaceship, when we put it into the patient, will find its way into the cells that it needs to go. The other type of gene therapy is called ex vivo gene therapy, which is where we take cells out of a patient who has Batten disease, and then we use that vector or spaceship to put the gene into the cells, and then we put the corrected cells back into someone's body. So the major difference basically is at what point does the vector or the rocket ship get into the cells? Is it inside the body or outside the body? Most of the gene therapy research in Batten disease is in vivo, which is using a kind of virus called an AAV virus to package the gene and get it into the cells. So the rocket ship is that AAV virus. In addition to enzyme replacement and gene therapy, there are also therapies that involve small molecules that can be given either by mouth or intravenously through the vein. There are also some large molecule therapies that work by altering the immune system. Some therapies involve molecules that can get into the brain themselves, and some we have to put in the brain directly. At this point, we don't know exactly for sure which type of approach is going to be best, and likely the best benefit will come from a combination therapy rather than any single therapy. If you're interested in hearing more about ongoing research in gene therapy for Batten disease, stay tuned for a few talks coming up during the conference. Dr. David Pierce will do an overview of ongoing research in Batten disease and different therapies that are in the therapeutic pipeline. And then we'll have updates from 20 different researchers on their projects, along with a live Q&A session with Dr. John Cooper. In summary, the things that we talked about today. So we learned that Batten disease is a name to describe a group of diseases called neuronal ceroid lipofuscinoses, or NCLs, which are lysosomal storage disorders. We learned that the lysosomes are the part of the cell that breaks down waste products. In Batten disease, a specific waste product called ceroid lipofuscin builds up inside cells of the brain. We learned that Batten disease is a genetic disease that happens when you don't have at least one functioning copy of the gene. There are 13 different forms of Batten disease with many things in common and many differences. And finally, there are 3 different types of treatments that we discussed today, which were symptomatic care, enzyme replacement therapy, and gene therapy. Thank you so much for coming to this talk and listening today. I would just like to give a huge thank you to the Batten research team at the University of Rochester, whose names are on the screen here, as well as a huge thank you to Dr. John Mink and Margot Maston, who contributed materials and helped me put together this presentation. Thank you so much. Bye-bye.